Hello everyone, my name is Min from McGill University, and my topic is novel polyfluoroalkyl betaines persist in oxic soils and bioaccumulate in earthworms. As we all know, aqueous film forming forms AFFF can be used to extinguish hydrocarbon-based fuel fires due to the presence of fluorinated components inside. The PFAS compositions in HFF evolved with the phase-out of PFOS, PFOA, and longer-chain perfluoroalkyl acids. Specifically, the historically manufactured HFF contains perfluoroalkyl sulfonates or ECF-based precursors as important components, while the more recent HFF primarily comprise NO2 fluorotenomers. There are studies that reported that certain currently in use HFF with the brand name of Ansu or Bakai contain NO3 and over1 or 2 fluorotenomers as significant components. Such kind of HPR use have resulted in the wide occurrence of these novel fluorotemer betaines with N over 3 and over 1 over 2 polyfluoroalkyl chains in the environment and biowater, including soil, surface water, sediment, and fish. ECF-based precursors manufactured via electrochemical fluorination was found to be able to degrade into perfluoroalkyl sulfonamines as transformation intermediate, which can further break down into perfluoroalkyl sulfonates as terminal products. In contrast, the NO2 fluorotemer manufactured via telomerization process was found to be able to degrade into NO2 fluorotemer sulfonate as transformation intermediate, which could further break down into a series of perfluoroalkyl carboxylates. The NO3 and NO1 over 2 fluorotemer betaine compounds was manufactured via different process, possibly through hydroxylation of unsaturated polyfluoroalkyl amine. The different chemistry might enable this compound to exhibit different fate and behaviors after release into the environment. This aspect has never been studied before. To fill these knowledge gaps, our studies aims at investigating the biotransformation potential of those novel fluorotemer betaines in aerobic soils and exploring the bioaccumulation potential of those compounds in earthworms. For the biotransformation experiment, soils were specced with either pure fluorotemer betaine compounds or fluorotemer betaine containing HFF and incubated for an extended time period. At select time points, both soil and headspace samples were collected and analyzed by high-resolution mass spectrometry with the use of target, suspect sequencing, and non-target analysis for checking the concentration change in both parent compound and the potential transformation product. When the soil was specced with 5 over 3 fluorotemer betaine compounds, the concentration of the parent compounds in both live and sterile cells show little or no change over time, and simultaneously, the 5 over 3 fluorotemer carboxylate were sporadically detected in the live soils at 2 time points. The carbon 3 and carbon 4 PFC, that is PFPR and PFBA, remain undetectable or at trace levels in the live soil. The carbon 5 and carbon 6 PFC, that is PFP and PFHEX, didn't show clear trend in live soils, despite their slightly higher levels in the live soil compared with the sterile and the live soil control group. And these findings exclude the conformation of this short-chain polyfluoroalkyl acid and short-chain perfluoroalkyl carboxylates as biotransformation products of 5 over 3 FTB. These 5 over 3 FTB compounds contain only 5 fluorinated carbons. So the PFAS with 6 or more fluorinated carbons are improbable transformation products. And the experimental results did confirm this. The concentrations of the carbon 7 to carbon 11 PFC remain consistent in all 3 soil groups. And also the concentration of the PFSA with 6, 8, or 10 carbons also remain constant in the 3 soils. But surprisingly, we did observe a continually increasing trend for perfluorobutane sulfonate, that is PFBS, and also perfluorobutane sulfamide, that is FBSA, in the live soil, but not in sterile and live soil matrix control. And this FBSA and PFBS were supposed to result from the biotransformation of carbon-4 ECF-based precursor rather than the parent 5-over-3 FTP. And third precursor were not to be present in the soil matrix background and might be introduced during the incubation experiment. To conclude, the biotransformation of 5-over-3 FTP compounds occurs slowly in Arabic soils, with little or no formation of short-chain polyfluoroalkyl acid and short-chain perfluoroalkyl carboxylates. When the soil incubated with 5-over-1 or 2 FTP compounds, the concentration of the compounds in both live and sterile soils showed negligible decline in the concentrations over time. And simultaneously, the production of 5-over-3 FTCE and carbon-3 to carbon-6 PFCs were insignificant. And these findings support the conclusion that 5-over-1 or 2 FTP compounds is not readily available in Arabic soil. When this soil were incubated with FTP containing H5F, we found that the parent N1 or 2 FTP and N3 FTP compounds showed a negligible decrease throughout the incubation period, including both the short chain 5-over-3 and 5-over-1 or 2 FTP and the longer chain FTP with 7 or 9 or 11 carbons. And simultaneously, the concentration of 5-over-3 FTC shows 5 to carbon-9 PFCs showed a fast increase in live soil compared with the sterile soil, and those findings confirm the production of these polyfluoroalkyl acid and both short chain and long chain PFC from the biotransformation of this H5F. By day 120, the concentration of carbon-5 to carbon-9 PFC reached 0.005 molar percent to 0.028 molar percent, and the total PFC concentration amounted to 0.07 molar percent in the live soil. 3 other soils were also specced with the same isolate H5F, and to examine the variations in the biotransformation outcome. And the time profiles and trends of the H5-derived PFAS were similar in 3 soils, and also the simultaneous production of carbon-5 to carbon-9 PFCs were similar as observed in the above-mentioned soil. By the 120, the concentration of carbon-5 to carbon-9 PFCs was slightly higher compared with the above-mentioned soil, and the total PFC yield in these 3 live soils were slightly higher compared with the above-mentioned soil. So in conclusion, the incubation of a commercial H5 in 4 soils resulting in the production of 0.07 to 0.40 molar percent PFC by day 120. The left map shows the different geologic locations for the collection of the 4 soils used for the biotransformation study. The right figure for the MDS plot demonstrated the distinct microbial communities in the 4 soils. And despite the different physical chemical properties and also the distinct microbial communities in the 4 soils, the outcome of H5 biotransformation were similar, all showing persistence of this H5 in these surface or subsurface soils. The non-target analysis and the suspect sequencing analysis were also performed to examine the production of other potential transformation products. And only one compound, that is 5-over-3 fructanomer methylamine compound, were identified to be transformation products product of 5-over-3 FTP, since its concentration showed an increasing trend followed by a decreasing trend over time. For the bioaccumulation experiment, the laboratory growing earthworms were exposed to either an airport soil impacted by HVF or clean soil specked with pure PFAS compound or HVF mixture. And after 21 days of uptake phase, the soil and earthworm samples were collected and analyzed for the PFAS concentration to get a soil earthworm bioaccumulation factor. This figure shows the bioaccumulation factors in the spec soils. As we can see that the OR3 and OR1, OR2 fluoroalkyl compounds showed a bioaccumulation factor comparable to that of short-chain PFCs, but lower than that of longer-chain PFC with 9 and 10 carbons, and also much lower than that of polyfluoroalkyl sulfonates with 4, 6, and 8 carbons. And additionally, we found that the biotransformation potential of these fluoroalkyls were related to the polyfluoroalkyl chainlets, with the highest bioaccumulation factor observed for the 11/3 and 11/1/2 FTB. In the airport soil impacted by HVF, similar trends were noted, and 1/3 and 1/2 FTB compounds were less bioaccumulative compared with polyfluoroalkyl sulfonates and longer-chain polyfluoroalkyl carboxylates. In the field soil and earthworm collected at a major Canadian airport site, the field-derived bioaccumulation factor of OR3 and OR1/OR2 FTB compounds showed lower bioaccumulation potential compared with the longer-chain TFCA with 9 carbon and 10 carbon, and much lower than that of polyfluoroalkyl sulfonates with 8 carbon. And also we noticed that the field-derived bioaccumulation factor were 10 to 15 times larger compared with the experiment, compared with the laboratory-derived ones. But still, we found that the OR3 and OR1, OR2 FTBs showed higher body burdens in these field earthworms, along with other HVF-derived components such as PFOS. And this was supposed to be due to the reduced biotransformation capacity of these telomeres in the invertebrate species earthworms. So to conclude, 5/3 and 5/1/2 FTBs or FTB containing HVF were highly persistent in Arabic soils. At best, it can only produce trace amount of short-chain polyfluoroalkyl acid and short-chain polyfluoroalkyl carboxylates. In addition, both the laboratory bioaccumulation test and the field observation data suggest that the OR3 and OR1/OR2 fluoroalkyl showed a lower bioaccumulative potential compared with the highly bioaccumulative polyfluoroalkyl sulfonates, but still remain moderately bioaccumulative. The present work represents the first study that demonstrated that these novel fluoroalkyls are persistent in soils and also in vertebrate species. I'd like to thank my supervisors, Professor Jin Xialiu and Professor Sebastian Sauvé, for their excellent guidance and tremendous support in this project. And also, I would like to express my great appreciation to Dr. Gabriel Monas and Dr. Song, and also our lab group members and external collaborators for their great support. And lastly, I would like to thank the great funding support from NS, ERC, and CDEP. Thank you, everyone, for your attention.